Right. Hi, everyone. So welcome to our class. So today we are just going to do a bit of corrections on the exercise that you have done before. And the exercise was the one that I posted on Go Formative, and that was on plant nutrition. Okay. And I'm not going to go through every question. So I'm just going to go through the questions that most of you actually got it wrong. Okay. So without further ado, okay, let's just jump straight to it. So the first questions that most of you struggle with was actually this question number 2. Okay, so let's just go through the questions first. Okay, so if you see here, it says a plant has leaves that are green in the center but white around the edges. A plant is starch and then has one of its leaves partly covered with black paper on both sides of the leaf as shown. So what you have is you are presented with this diagram here, right? And they actually told you that you have the green area and you have the white area on the outside, okay, on the outside surface. And what they have done was they actually place a piece of black paper in the middle. So this is actually to block light. So let's just put our annotations here. So black paper to block light from passing through. Okay, so and you have white area of leaf and green area of leaf. And you should know that if it's green area of leaf, so which means it automatically means that it has chlorophyll. Okay, and you should know by now, so whenever they have chlorophyll, that means they are involved in photosynthesis. Okay, and what is photosynthesis? Just for us to review back. So photosynthesis is essentially it's CO2. Sorry, let's just find pen. Yes, so CO2 plus H2O in the presence of light and chlorophyll will give you glucose plus oxygen. So glucose is C6H12O6. H12O6, right? And you should know by now as well, now this glucose, what will happen is any of the excess glucose, they will be converted into starch, stored as starch, okay, which is essentially a form of carbohydrate. Right? So now, again, just going through, you have the leaf. Okay, in this type, this is what we call as variegated leaf. Variegated leaf. And variegated leaf, it has white area and green area. So you have the black paper for what? To block light from passing through. White area, green area has chlorophyll. So which means if light is present here, that means they will have photosynthesis occurring. And if they have photosynthesis, that means there will be starch here. Okay, so in this case, green area involved in photosynthesis. So starch, yes. Whereas if it's white area, that means no chlorophyll. And if it's no chlorophyll, that means no photosynthesis. And if it's no photosynthesis, it means starch will also be absent, right? And it says here, the plant is placed in bright light for 48 hours and the leaves then tested for starch. This diagram shows the colors that are obtained. And if you test for starch, okay, if you test for starch, you are actually using iodine. Okay, you use iodine solution, and you should know as well that if it's iodine, if it's a positive result, so which means if starch is present, so they will actually turn blue-black. Whereas if iodine is absent, if starch is absent, then it would remain yellow. Remain yellowish brown. Okay, so this is the ones that you should test. And from here, you should know by now, okay, if you test these areas with starch, okay, with iodine solutions, right? If you test it with iodine, so what will happen, okay, based on the information that we gather here, okay, the ones that we have. So which means, okay, so you have the strip here will block light. So if light is not passing through here, so whether it's green, whether it's white, so they will have no photosynthesis regardless, okay? And if no photosynthesis, that means it would be no starch. And if it's no starch, so which means it would be negative. So what will happen? They will remain yellow. So where would the yellow be? Okay, the yellowish brown, it would be tanga-tanga. Okay, so the ones that you should have is either A or B. So C and B is confirmed salah sudah. Okay, it's confirmed salah sudah because it says here yang tengah-tengah yang bertutup itu, it's black cakap here. Okay, and it will only turn black kalau misalnya starch is present. But in this case, starch is not present. Okay, in this case here, no chlorophyll, no photosynthesis, no starch. No starch means iodine solution, negative, yellow. Right? So here it should be yellowish brown, yellowish brown. Okay. But the question is, which one? Is it A or D? Again, if you look here, yang siring-siring, okay, yang siring-siring on the outside is actually white. And if it's white, so which means no chlorophyll. And if it's no chlorophyll, no photosynthesis, so no starch. And if it's no starch, it should be iodine solution, negative. So it should remain yellow. So in other words, the outside should also remain brown or yellow. Okay, remain yellowish brown. So which means this is salah, right? So your answer should be A, right? So this is how you should do, right? Next question that I will be discussing is actually question 5, right? So question 5 is this question here, right? So if you look here in question 5, it says a container is filled with water from a lake and placed on an outside table in the sunlight. Healthy plant seedlings are placed in the container and left for 3 months. Distilled water is added to the container as required. At the end of each month, a typical mature leaf grown during the previous 30 days is removed and drawn to the same scale. And the diagram shows 3 drawings that are made. Okay, so this is month 1, month 2, and month 3. Question is, why is the leaf from month 3 is smallest? Okay, so you have, it says here, a container filled with water from a lake, placed outside in the sunlight, and then left for 3 months. Distilled water is added, and at each end of the month, the previous 3 days is removed and is drawn to the same scale. Okay, so here, what you have is actually this. Okay, So you have a lake water, awal-awal. Okay, awal-awal, you have it in lake water. The plant was originally grown in lake water. So before, they were grown in lake water. Okay, and if it's grown in lake water, so which means they have all the nutrients naturally available. Okay, because it is lake water, right? So all the nutrients, they would be naturally available from the lake anyway, right? And once you took them out, once you took it out, what they have done is they actually replace it with distilled water. Okay, so they took it out. So after, they will replace this. It's actually grown in distilled water. And what you should know is if it's distilled water, it simply means pure water. Pure water, so in other words, there is no nutrients present. So there is no nutrients, there is no minerals. Okay, there is no nutrients, there is no minerals, so there is no nitrate, no magnesium, so on and so forth. Okay, and you should know that nutrients are needed for plant growth. Okay, so in other words, if you don't have nutrients, it would mean there would be less growth. Okay, and if it's less growth, of course, the longer you leave it, the smaller it will get. Okay, so here, why is the leaf for month 3 is the smallest? Kenapa ani paling lambat cakapnya? It's simply because insufficient light? No, because they actually always give sunlight. Okay, if we look at C, phloem vessels unable to transport water? No, because they don't take out the phloem. Okay, and D, plants cannot grow well without soil. Not necessarily, right? Because you actually, the soil is just there simply because it has nutrients, right? But even if it's in water, if it has the nutrients, then it can grow well without the soil as well. Okay, so the only possible answer is B. Okay, nitrates from water has all been absorbed. So which means all the nitrates yang available from the lake water, it has been absorbed. So now they replace it with distilled water. So no nitrates, no mineral, no growth, which means it will get smallest here. Okay, so that's why the answer is B. Okay. Oh, by the way, I actually forgot about this question as well. Okay, if we go to this question. Yes, this question is similar to the question that we discussed earlier. So it says here, a variegated plant is de-starched and then placed in sunlight for several hours. The leaf is then taken and tested for starch using iodine solution. So again, okay, so you have variegated plants. So variegated plants, which means it has the white plus green. Okay, so you have the white plus green. So that's what they show you here, right? But this time, they didn't give anything in between. They didn't give anything in the center. It's simply just, they take out this leaf, they de-starch it, and then afterwards, they place it in sunlight, and then afterwards, they test it with iodine. Okay, so you should know now, if it's green, that means chlorophyll. Chlorophyll is present. And if chlorophyll is present, that means yes, photosynthesis. And if it's yes, photosynthesis, that means yes, starch. And if it's yes, starch, if you test it with iodine, then they will actually give positive results. If it's positive, then they will actually turn blue-black. Okay, around the green area. So green, chlorophyll is present. If chlorophyll is present, that means they will have photosynthesis occur. If photosynthesis occur, that means you will have storage of starch. If you have storage of starch, then if you test it with iodine, it will actually turn blue-black, right? So all the green part, it should become blue-black. Whereas if it's white, okay, if it's white, that means no chlorophyll. If it's no chlorophyll, that means no photosynthesis. If there is no photosynthesis, that means no starch. If there is no starch, if you test it with iodine, then it would be negative results. And what will be the negative result? It will actually remain yellowish brown. Okay, so all the white, it should remain yellowish brown. All the green, it should turn blue-black. Okay, and obviously, which one would be the answer? If you look, okay, the answer is quite obvious. The answer should actually be C. Okay. Now, next question. Next question is question 9. It says here, a growing plant is regularly watered with a solution. The composition of solution is changed after which the plant leaves become yellow in color. Okay, so for this question, right? So for this question is actually, oh wait, is it this question? No, sorry. It's actually question 10, right? So question 10, not question 9. Right? So if we go through question 10, it says here, the diagram shows a green shoot photosynthesizing under a glass jar. This was used as a control experiment in a lab investigation. So what you have is you have a plant, okay, and it's green plant. So automatically you can assume that there is chlorophyll. So yes, chlorophyll. Okay, and before that, we just go through photosynthesis. Just remind ourselves photosynthesis, the equation for photosynthesis. So it should be CO2 plus H2O will in the presence of light and chlorophyll to give C6H12O6 plus O2. Okay, so just quickly to remind ourselves, this is the word equation and this is what has been given. Okay, they have actually give you green plants. So yes, photosynthesis. They actually give you sunshine, so which means light is also present. Okay, so light is present. And they give you, even though it's airtight, but you could assume that there is air here. So which means if it's, even though it's airtight, so that means there is still air inside. Okay, they just trap the air in, right? So you can assume that there is carbon dioxide available. And of course, they also have water. So there's also water. So in this case, in the control experiment, okay, in the normal setup, there will be actually photosynthesis taking place. So if this, all of this is met, so that means photosynthesis. So that means photosynthesis is occurring, right? And this is in the setup of the control experiment. Bear with me for a second. Right, so in this case here, it says which diagram shows the experiment carried out to investigate the need for carbon dioxide in photosynthesis, right? So which diagram shows experiment carried out to investigate the need for carbon dioxide in photosynthesis? Now, if we want to investigate the need for carbon dioxide, okay, investigate the need for carbon dioxide. So which means if you want to know whether something is important, so if you want to know something is important, what you will do is you need to remove it first. Okay, so as I mentioned in the video last time, okay, kalau kamu kan tahu orang itu kamu sayang berhabis kena, so break up dulu. Kalau misalnya masih kamu ingat-ingat, artinya kamu berguna berhabis-habis. Okay, so same thing here. If you want to know, you want to investigate the need for carbon dioxide in photosynthesis. So what you should have here in this investigation, you should actually remove the CO2. Okay, remove the CO2 in order to know its worth. Okay, so if you want to investigate whether it is important, so what you should do is you remove that factor. Okay, so again, as I mentioned, kalau kamu kan tahu someone itu important arah kamu, let's say ia hilang. Kalau ia hilang, lepas itu kamu tidak dapat buat apa-apa, so which means ia tu is important arah kamu. So same thing here. So carbon dioxide, kalau kamu removekan, something is happening arah the photosynthesis, tidak kita buat apa-apa photosynthesisnya, that means CO2 is important. Okay, and if you look here in this setup, okay, so you have these four setups here. Okay, so which means of all of these four, we need to know. So what we should do is find which setup removes the CO2. Okay, so this is what we should find, right? Because we want to investigate the need for carbon dioxide. Okay, this is what we are investigating. We want to know carbon dioxide is important. So if you want to know this, that it is important, so we remove it. So in other words, we need to find which setup removes the CO2. Okay, and I've mentioned in the video before that if you come across, okay, remove the CO2, it could be removed the CO2 macam mana. So number one is obviously if it's in a vacuum. Okay, if they place it in a vacuum state, so no air at all. So if it's no air, that means no carbon dioxide. Or number two, if that actually passes through or if they include these solutions. So what are the solutions? Sodium, ataupun potassium, ataupun lithium hydroxide. Okay, so if they include sodium hydroxide or potassium hydroxide or lithium hydroxide, if they include all of these three, okay, or any of these three, what all of these will actually do, they are absorbing the CO2. Okay, so absorbing the CO2, so in other words, it removes the CO2 from the air. Okay, in this case, it removes it from the air, right? So if it removes the CO2 from the air, so as a result, so photosynthesis will not actually occur, right? So in this setup, which one removes carbon dioxide? The only setup which removes carbon dioxide is actually A, because it has sodium hydroxide solution. Okay, so this sodium hydroxide removes CO2 from air. Okay, if you see here, hydrogen carbonate indicator, it's simply just an indicator. Okay, indicator, you want to know CO2 is present or not present. Bukannya to remove. Okay, this is just an indicator. Okay, indicator is simply you want to know whether it's there or whether it's not. It's not trying to remove it. Okay, the only thing that removes carbon dioxide is this. So that's why the answer is okay. Right. Next question is question this one. Oh, okay. It says which graph shows the effect of temperature on the rate of photosynthesis. Now, as I mentioned before, that anything to do with temperature is actually to do with enzymes. Okay, and if it's to do with enzymes, you just simply recall back the graph, okay, of all of these, which of these graphs actually fits the effects of temperature on enzyme activity. And all of these, okay, from all of these four, the one that actually shows the effect of temperature on enzyme, the one that fits the most is actually C. Okay, so if you have trouble with this, please refer to your enzymes in your graph. Okay, the effect of temperature on enzymes in your graph. Okay, it should be familiar to you. Now, the next question is this. Wait up. Right, so the questions that I will be discussing. So it says question 14. A student investigates the effect of different colors of light on the rate of photosynthesis. In 3 separate experiments, he shines red, blue, or green light onto an aquatic plant, and the number of oxygen bubbles produced by the plant is counted. Each experiment is carried out 3 times, and the average number of bubbles calculated, right? So you have here, right? So what you have here is you have 3 different light sources of different colors. So you have red, blue, and green, right? If you see here, if you provide the plant, okay, so you have different on the rate of photosynthesis. If you have plant, you provide them with red, they will produce 48 bubbles in 1 minute. If you give blue light, it will produce 37 bubbles per minute. And if you actually give green light, it will actually produce 12 bubbles per minute. So if you see here, the more bubbles that are produced, it simply means more photosynthesis. So more bubbles, it simply means more photosynthesis. Okay, so more bubbles, more photosynthesis. If it's less bubbles, it would mean less photosynthesis. Right? So now you might be asking, what's the relationship between bubbles and photosynthesis anyway? Well, if you recall photosynthesis, okay, so if you recall photosynthesis, it's CO2 plus H2O will give C6H12O6 plus O2, right? So they absorb carbon dioxide, which is in the gas form. They absorb water, which is in aqueous form. They will produce carbon dioxide, sorry, they will produce glucose, which is in aqueous form. And they will
oxygen, which is a gaseous form. So the bubbles that are actually produced is actually not because of this. It's actually because of the oxygen that are being produced. Okay, so that's why it actually relates to here. Okay, so photosynthesis, it will give out oxygen, and that oxygen, it will produce the bubbles because you put the plants, you submerge the plants with in water, because it says here aquatic plant. Okay, so aquatic plants, that means you leave them in water, right? And if you leave plants in water, so of course the oxygen will simply bubbles off, right? So the more bubbles that you have, more photosynthesis. Right, so what's the questions then? If you see here, what explain the results? Okay, so if you look here, we can see red color produces the most bubbles, which means most photosynthesis. Green color produces the least amount of bubble, which means it has the least photosynthesis. And blue color is the medium amount, so therefore it is the medium photosynthesis. So it says what explains the results here. It says here, if you look at the conclusions, so it says here, chlorophyll absorbs red and blue light more than green light. So is that true? Red and blue, the photosynthesis, they absorb red and blue more compared to green. Is that true? Yes, because red is more than green, whereas blue is also more than green. So which means red has more photosynthesis than green. So same thing, blue has more photosynthesis than green. So chlorophyll absorbs red and blue more than green. So that is the answer. Okay, so in this case, the answer is actually A. Okay, so it says here, B, green light is absorbed by water. No. Why? Because they don't say anything about green light being absorbed by the water. There is no data telling you that the green light is absorbed by water. Okay, so that's why the answer is not B. C, most of the green light is absorbed by the chlorophyll, which is not supported. Okay, because if it's absorbed by the green, if the green is actually absorbed, if they absorb, that means they will produce photosynthesis, they will produce bubbles. So in this case, if you say this is right, so your answer should be, this one would be the highest. But in this case, this is not the case. So therefore, C is wrong. And D, red light is least in photosynthesis. Again, this is wrong as well. Because from here you see red is the highest. So highest means more photosynthesis, right? The least is actually green. Okay. The next question is actually this. Right. In an experiment is set up as shown. The volume of gas collected is measured after 30 minutes. So here you have aquatic plants. That simply means plants that live in water. Okay, so they live in water, they live underwater. And so we can assume that they actually has chlorophyll. So which means that they are green. Okay, and the gas being produced is, as I mentioned earlier, this is to do with photosynthesis. So if we just go back to word equation, okay, sorry, the chemical equation. So it would be CO2 plus H2O, light, and chlorophyll will actually give out C6H12O6 plus O2. And this O2 was actually the bubbles that are being produced. right? And you have light source there as well. And inside the water here is, you can assume that there is actually dissolved CO2. Okay, so we can assume that actually has dissolved CO2 because it's not covered, right? And they don't put any sodium hydroxide in, right? So that's why this is. Now, the experiment is repeated several times. Each time the light intensity is increased. Okay, which graph shows the results, right? So in other words, this is actually asking you, okay, the question is asking you, right? The question is simply actually asking you, which graph shows effect of light intensity on rate of photosynthesis. Okay. Yes, I know it says here volume of gas, volume of gas, volume of gas. But as I mentioned earlier, it is the volume of gas, which means the amount of gas collected. So the more gas you have, if you have a lot of gas collected, it simply means a lot of oxygen. If you have a lot of oxygen produced, it also means that there is a lot of photosynthesis occurring. Okay, so the more gas you collected, that means more photosynthesis, right? And so that's why this is similar to this, right? So which graph shows the results? So it's simply asking which of the graph is showing you the effect of light intensity on the rate of photosynthesis, okay? And I have mentioned in the previous video that of all of these, the graph that actually fits this is actually graph C. Okay, so if you are in doubt, please rewatch the video that I posted before. Right, so next question is this. Right, the diagram represents the cross sections of part of a leaf. So this is the cross sections of a leaf. If I cut through a leaf, this is what I would get, right? If you just label them first, so you should be able to label them. So the top part is the waxy cuticle. This whole layer of cells here, all these 3 cells here, is actually the upper epidermis. And then you have all of these cells. Okay, these cells arranged, the long cylindrical cells. These are the palisade mesophyll. And the cells, the irregular shaped cells, are what we call as the spongy mesophyll. Right, and here you've actually got these cells. Those are actually your vascular bundle. Right, so vascular bundle, which consists of xylem and phloem. And the lower cells here is your lower epidermis. And yeah, so lower epidermis. But what is not shown here is they don't show here your gut cells. Okay, the gut cell together with the stomata. Okay, it's actually not shown. Okay, but you should be able to know that it is there. Right, so the question is asking, how does the oxygen content of the air at X compare to the normal atmospheric air when the leaf is in the light and when it is in the dark? So when it's at X, so at X is actually the normal cells. Sorry, X is your intercellular airspace. Okay, so if your X there is airspace, right? So how does the intercellular airspace differ from the atmospheric air from the outside when it is in the light and when it is in the dark, right? So we want to know the oxygen content. And you should be able to know that in the light, when there is light, so that means yes, photosynthesis. And if they actually do photosynthesis, that means yes, oxygen. Okay, so they will actually produce a lot of oxygen. And this oxygen produced, it would be in the airspace. Whereas in the dark, what will happen is no photosynthesis. And if they don't have photosynthesis, that means less oxygen in airspace. Right? So based from here, right? So automatically you should be able to decipher what's the answer. So the answer in the light, yes, photosynthesis, yes, oxygen. So if we compare with the atmospheric air, so during the day, the intercellular airspace should actually have higher oxygen, whereas in the dark, it should be lower, simply because it has no photosynthesis. So the answer is A. Right? Then the next question. This one. A student performed an experiment to see whether chlorophyll is necessary for photosynthesis. What is a suitable plant for this experiment? So again, they want to know whether chlorophyll is necessary. So if you want to know chlorophyll is necessary, kalau kami kan tahu chlorophyll itu berguna ke indah, so what we should have done is we remove the chlorophyll. There. So which means, in other words, you choose the plants that has no green. Okay, and a plant which has no green area, so it's in other words, it's asking you to use the variegated leaf. Okay, this is actually the best leaf that you can use if you want to test the importance for chlorophyll in photosynthesis, right? Simply because if you use variegated leaf, it means that there would be no chlorophyll because it has no green area. So yeah, because it has white area and green area. Right? So from there, you can actually use that leaf, the variegated leaf, to see whether chlorophyll is necessary. Right? So all of these options, among all of that options, the ones which are okay, so among all of these options, the ones which makes the most sense is B. Okay, a plant which has white patches on its leaf. Okay, no green areas. In other words, white patches. Okay. So all of these, the answer is actually B. Right. Then I think we are getting to the quite close to the last questions. Here, this question. It says the diagram shows some of the structures seen in a section through dicotyledonous leaf. So this is your leaf structure, which two features are missing from this diagram. So you have, as we recall this part earlier, So if we simply just recall and we compare, so you can obviously see that what is missing, there is missing the vascular bundle, and together with this as well, they don't actually show you the gut cell, the stomata, right? So here, what is missing? Okay, the features that are missing, not present in the diagram, it would be gut cells and vascular bundle. So the answer is actually C. Right. This one, similar. Okay, it's similar questions to what I have shown. Okay, what we have discussed is they want to investigate the need for CO2. So if you want to know the need for CO2, that means you need to have experimental setup. Experimental setup that eliminates or removes CO2. I have told you that sodium hydroxide, anything sodium hydroxide or potassium hydroxide or lithium hydroxide, they will absorb the CO2. That means no CO2 here. If there is no CO2, then that is what the one that they investigate. Okay, and it's actually mentioned in the question as well, right? Sodium hydroxide absorb the CO2. Okay, so that means all the CO2 here gets here instead, masuk dalam the sodium hydroxide, not into the plant. Okay, so obviously the answer is A. And I think which one is question of this question? So this question is the last question. The graph shows how the rate of photosynthesis varies with light intensity to different temperatures. Other variables are kept the same. So you have light intensity and you have temperature as well. So you actually have two factors considered here, right? So this is to do with limiting rate of photosynthesis. So in other words, it's actually asking you about limiting factor. Okay. So just to recall back, as I mentioned from the previous video, that limiting factor, what does it mean? Any factor in which if you change it, it will also change the rate of reaction. Okay, so if you, limiting factor is any factor, if you change it, it will also change the rate of reaction. So for example, if you increase light, if you increase the light and somehow the rate of photosynthesis also increase, that means at that point, okay, at this point here, light is limiting. Okay, so as we know for sure that light intensity is limiting. Why? Because if at this point at S, if I increase the light, the rate of photosynthesis also increase. So I know that S is definitely light is limiting. Okay, limited by light. Okay, the same thing with Q as well. So if you see here, this is the curve for Q. Okay, so if you see the curve for Q, if you increase the light, the Q punya curve also increasing. So same thing, it is also limited by light. Okay, but the case with R, okay, the curve here, the region R, if you focus on R, if you increase the light intensity, the rate of photosynthesis is still constant. So in other words, BR, light is no longer limiting. Okay, and if light is no longer limiting, other factor may be limiting. Okay, example, temperature. Okay, how do I know? In this case here, this is the curve when it is 15, but at the same light intensity, if I increase the temperature, once I increase the temperature, suddenly the graph, the rate increases at the same light intensity. So here, what I can say is light plus temperature is no longer limiting. Okay, something else must be limiting. So other than that, something else, it could be chlorophyll. Okay, other factor, maybe okay. So going back to the question here, it says which sections of the graph is light intensity limiting? So which means which of these area in which if you change the light, it will also change the reaction. And that is actually to do with curve S, which is this curve, as well as curve Q, which is this curve here. So the answer is Q and S. So the answer is actually B. All right. So I think that's it for the corrections. So I will you should be able to answer the rest of the questions now. Okay, I will post another questions on Go Formative, and you should answer the questions there. Okay, so for now, I will leave it out, and I'll see you guys next time. Bye.